Now that we've learned about the cell cycle, we're going to learn about the different steps in mitosis. So here you have your interphase, and this is when everything is ready. This is the last stage of interphase. Everything is ready to begin mitosis. So these are the stages of mitosis. So you can see the cell is getting ready to divide in cytokinesis. Okay, so there are a series of steps that have to happen before your cell is actually ready to divide. And we're going to learn each of those steps. So along with cytokinesis, mitosis is part of the M phase. So before we had interphase with the G1, S phase, and G2 phase. Now, mitosis and cytokinesis are part of M phase. So this is when the chromosomes are going to be separated. So we can actually divide that cell into two cells. Remember, each of them will need their own set of chromosomes. This actually happens very quickly or very rapidly because when you are separating the chromosomes, we need to protect that DNA and make sure that we don't make any mistakes and nothing happens to that DNA. So this is going to happen quickly. The chromosomes are all molecules in the nucleus, and they contain our genetic material. So every organism has DNA in each and every cell. And the chromosomes are what we call the DNA. So they are arranged in chromosomes. So before mitosis, the chromosomes cannot be separated or seen easily. It looks like a bowl of yarn trying to figure out where the string ends and the other begins. So if we took a bunch of pieces of yarn and we cut them into sections and then we mix them all up together, it's really hard to tell where one starts and one begins. Also think about if you're eating like a bowl of spaghetti, okay? Yes, you might be able to see a couple of ends, but really it's all mixed together. You can't very easily separate all of the pieces just by looking at that bowl of spaghetti. That's kind of like your chromosomes. So whatever works for you, yarn, spaghetti, think of one of them. If you can think of another analogy, you're welcome to use that. And if you do think of a different one, I'd love for you to share. So before S phase, there are 46 unduplicated chromosomes floating around in the nucleus. So this is before the DNA actually replicates. Okay, we have 46 unduplicated chromosomes. After S phase, there are still 46 chromosomes, but now they are duplicated. Okay, so before I mentioned that we are doubling the DNA, but we're not actually doubling the number of chromosomes. So now they float around together, okay? They're with their partner, and we call them sister chromatids, and they are twins. So every chromosome now has a buddy, and that buddy is its twin. And they are held together at the center by a centromere. So this is a portion of the chromosome that attaches each of those sister chromatids. So we have multiple stages of mitosis, just like we had multiple stages of interphase. The first phase is prophase. So this is the before, okay? The chromosomes begin to become condensed, okay? They are going to really shrink in size. As they continue to do this, they take on that X shape. So here we have our X shapes, okay? This is one chromatid here, if you're looking at my mouse. This is the sister chromatid, and it's held together by a centromere. Think center, right? Centromere. So this is a chromatid, and this is the sister chromatid. So we still have the same number of chromosomes, we just have a copy, and they're still held together, okay? So in prophase, the nuclear membrane, remember that nucleus has a membrane all the way around it, it's going to start to break down and to disappear. And this is because the DNA needs to be separated. So those sister chromatids eventually are going to be separated to prepare to divide the cell into two different cells. And if we kept that membrane around, they wouldn't be able to separate appropriately. So this membrane around the nucleus is going to start to break down so that these chromatids can travel freely. In an animal cell, the centrioles are going to begin moving apart as well. Plant cells don't have centrioles. These right here are my centrioles, okay? So those are gonna move to opposite sides of the cell. In a plant cell, they're going to use the cell wall instead of centrioles. So spindle fibers will begin to attach the sister chromatids at the centromere. So they're going to form like little strings that attach to the centromere of each of the chromosomes, from the centriole to the centromere. So next we have metaphase. So you can see all of these chromosomes are lined up. They have these spindle fibers from the centriole over to the centrosome, okay? So the sister chromatids line up at the center of the cell, and what helps them to do that is the spindle fibers. This is called the metaphase plate, okay? Then after metaphase, we have anaphase. This is the apart stage. So anaphase, apart stage. And I also kind of think of when you look at these chromatids and they're like folded in half almost, it kind of looks like the outside of an A, okay? So that's anaphase. So the spindle fibers will begin to get shorter and shorter, kind of like a fishing rod, okay? The line, when you caught that fish and you got what you want, you're gonna start reeling it in. So those spindle fibers are gonna start reeling in those chromatids, and that pulls the chromatids apart, okay? Once the centromere is split, the chromosomes move toward the opposite poles of each other. So these used to be twins and they were connected, and now they're pulled apart. Same with these. So each of the chromatids is going to be pulled to an opposite end of the cell. Then we have telophase. So now things are starting to look a little different. This is the end phase. So the chromosomes are going to start to uncoil. So remember, they got condensed and they made that really nice X shape. Well, now they don't need to be quite so condensed and organized. So they're going to start to uncoil once they get to their ends of the cell, and that nuclear membrane is going to form around them again. And then in cytokinesis, this is the part where the cells are actually going to divide. This is technically not part of mitosis, okay? It is its own thing. So the membrane is going to pinch inward and divide the cytoplasm between the two parts, and now we have two new cells. So all of the moving to opposite ends of the cell of the chromosomes and chromatids was so that we could then later divide the cell and form two different cells. So the two new cells that are formed from the original cell are called daughter cells, okay? Both cells are completely identical. They have exactly all the same genetic information as the parent cell. It is a copy of the parent. Unless, of course, there's something that goes wrong and there is a mutation. Each of the cells has the same number of chromosomes, and each chromosome has the exact same genetic information. So you can go ahead and watch this video. I will add the link for you guys if I need to. I'll find out. But here we have, these are actual real cells. And this is what each stage looks like. So in interphase, we have our cells are creating a copy of that DNA, okay? Then we have prophase, and that nuclear membrane is breaking down. In metaphase are all lined up in the middle, all those chromosomes. Then in anaphase, we're starting to split them apart. So you can see the chromosomes on one end here and the other chromosomes on the other end here. And in telophase, it's actually starting to redevelop that nuclear membrane, and it's going to get ready to pinch and divide into two cells during cytokinesis. So here are all those stages again that you can see, but you notice this cell is square, so this is likely a plant cell, okay? So you can work through each of those steps and see that eventually they become two different cells. And here we are again. All right, next up is regulation of the cell cycle. So you'll find out what happens if things go wrong.